Think about the last time you ate a sandwich. You chewed it, you swallowed it, and then you completely forgot about it. 20 hours later, your body has pulled apart every molecule, sorted the useful nutrients from the waste, and shipped the good stuff into your bloodstream. You never gave it a single conscious thought. We tend to think of the stomach and intestines as passive plumbing, just a series of muscular tubes squeezing food along. But regulating the exact volume, pH, osmolarity, and chemical composition of that food in real time is a massive computational challenge. Your brain isn't sitting there deciding which enzymes to release or which muscles to contract. The hardware solving this problem is the enteric nervous system, or ENS, a name for the network of neurons specifically governing the gastrointestinal system. This is a dense, interconnected web of 500 million actual neurons embedded right inside the tissue of your gut wall. To put that scale into perspective, your gut contains 5 times more neurons than your entire spinal cord. If you gathered every sympathetic and parasympathetic ganglion in your body into a single pile, your digestive tract would still outnumber them. This system operates using the same biophysical principles as the cerebral cortex in your head. It uses threshold logic to make decisions, relies on coupled electrical oscillators to keep time, and communicates via millisecond synaptic transmission. The gut is a distributed, self-governing biophysical computer running parallel to the brain. When food enters this system, it has to travel through 20 to 25 feet of intestinal tubing. The physical problem is how to coordinate the rhythmic muscular contractions, the peristalsis, that push this material forward, all without central instructions from the brain. The solution lies in specialized pacemaker cells called the interstitial cells of Cajal, named after Santiago Ramon Cajal, who spotted them in the late 1800s. These cells spontaneously generate waves of electrical depolarization. Internally, the release of calcium triggers specialized chloride channels to open. Negatively charged chloride ions flow out, shifting the cell's internal voltage and creating a tiny electrical slow wave. This voltage shift then leaks into neighboring cells through physical protein tunnels called gap junctions. The electrical current propagates outward from cell to cell, sweeping along the gut wall. The mathematical propagation of this slow wave follows the Cable equation, the formula derived by Lord Kelvin in the 1850s, to calculate how electrical signals travel through transatlantic telegraph wires. This chart shows varying electrical rhythms across different gut regions. The stomach maintains a steady rhythm of 3 cycles per minute. But in the duodenum, the first stretch of the small intestine, the tempo jumps to 12 cycles per minute to aggressively mix food. Because these cells act as coupled oscillators, the faster pacemakers upstream electrically entrain the slower pacemakers downstream. The faster rhythm dominates, creating a coordinated electrical wave that forces material in one direction. The interstitial cells of Cajal form an autonomous, self-sustaining electrochemical clock. They dictate the physical tempo of digestion without receiving neurological input from the brain. Layered over this electrical clock are the myenteric and submucosal plexuses. These are sheets of interneurons and motor neurons, the processing units of the ENS, sandwiched between the muscle layers of the gut wall. When a lump of food stretches the intestinal wall, it physically opens mechanosensitive ion channels in the membranes of local sensory neurons, causing them to fire an action potential, a rapid pulse of electrical energy. That signal triggers a split response. It travels upstream, commanding muscle to contract, and downstream, commanding muscle to relax. Enteric neurons algebraically sum dozens of these inputs. If the combined inputs cross a voltage threshold, the neuron fires. If not, it remains silent. This exact mechanism appeared in the first simple invertebrates millions of years before the first centralized brain appeared in the biological record. The ENS performs true threshold logic computation. It is an autonomous hardware board processing sensory data and executing precise motor responses, even when physically severed from central command. The gut's electrical signals interface with a chemical language of over 30 distinct neurotransmitters, the same molecular messengers found in the brain. This chart shows the distribution of serotonin in the human body. 90% of your body's serotonin, the chemical associated with mood and emotion, is produced and localized in the tissue of your gut. The gut also produces roughly half of your dopamine. This chemical reservoir is managed by specialized sensors called enterochromaffin cells, which line the inside of the intestine to taste chemical metabolites and detect toxins. These cells also respond to physical force. When food presses against the gut lining, the cellular membrane stretches. This physical pressure forces piezo-2 channels, proteins that sense mechanical force, to snap open, allowing calcium ions to rush inside. That sudden influx of calcium triggers the cell to release serotonin, which binds to receptors on adjacent nerve endings. In a fraction of a second, the gut physically converts a mechanical force into a chemical message, and then immediately back into an electrical signal. This local computation eventually connects to headquarters via a physical cable called the vagus nerve, which runs directly from the abdomen up to the brainstem. This diagram reveals the vagus nerve's bandwidth ratio. 80% of fibers are afferent, carrying sensory data up from the gut, while only 20% carry commands downward. These upward signals terminate in the amygdala, hippocampus, and prefrontal cortex, governing fear, memory, and decision-making. The old biological model suggested gut-to-brain communication relied on hormones slowly diffusing through the bloodstream. This was updated by the discovery of neuropod cells. Neuropod cells are gut lining cells that form direct, structurally complete physical synapses with the terminals of the vagus nerve. By firing glutamate, the primary neurotransmitter for fast signaling, across these tiny gaps, gut data reaches the brainstem in tens of milliseconds. Gut-to-brain communication is a high-speed neural hotline, feeding raw environmental data directly into your emotional centers at the speed of thought. Trillions of bacteria, viruses, and fungi in your gut microbiome also participate in this computational circuit. This readout compares the electrical activity of enteric neurons. On the left is a normal circuit. On the right is a circuit from a germ-free mouse raised without bacteria. Without microbial interaction, the enteric neurons fire sluggishly, and their electrical recovery period, the after hyperpolarization, is prolonged. The lack of bacteria physically changes the electrical recovery period of the neurons, proving the network is shaped by its microbial tenants. A single bacteria in your colon produces a metabolite. A sensory cell in the gut lining detects it and releases a neurotransmitter. A vagal nerve terminal catches that signal and fires an impulse straight to the brainstem. Microbes produce chemical variables that tune the electrical excitability of your internal logic gates. Gut bacteria provide the inputs that help modulate central cognitive states like anxiety and learned fear. We are looking at a 25-foot organ running on autonomous pacemaker clocks, threshold logic gates, high-speed synapses, and dynamically updated microbial inputs. The governing mathematics of thought are universal. The Nernst equation, which describes ion equilibrium, and Ohm's law, which governs current flow, apply identically to the tissue inside your skull and the tissue inside your abdomen. If we define a brain as a network that processes information using electrical and chemical signals, the gut meets every functional criterion. Your central nervous system relies on this localized, special-purpose computer to handle the overwhelming computational load of digestion. Without it, your brain would be too overloaded to focus on external survival. Gut feelings are literal, high-speed neural reports from your abdomen, evaluating threats and influencing your decision-making long before conscious awareness kicks in. The physical and functional connectivity between these two organs suggests that intelligence and emotional processing are not centralized, but distributed across the human machine.